I'm very thankful to the organizers for this opportunity to present the research going on in my lab right now, one specific project. I got quite ambitious with my title, and when I started putting my talk together, I realized there's no way I'm going to be able to incorporate all of that, so I've chosen a smaller portion of it. My lab focuses in general on the immunity, metabolism, and the microbiome, and today I'm going to talk about a project looking at the metabolic impact of immune response. So keep it simple. So there's no one here who hasn't experienced illness and all of the fun symptoms that go with it. It's actually a major drain on our productivity of our workforce that when we experience infectious disease, we lose time at work. We have fever, chills, lethargy, body aches, loss of appetite, sometimes weight loss. And this happens not only in humans, but in the animal kingdom. It's considered a very serious, it has a serious impact on performance and reproduction in animals. It's considered a resource trade-off between immune response and other activities, other life traits. So it is widely considered that the immune response to pathogens has a high metabolic cost, but we know isolated pieces of how fever impacts metabolism and how certain aspects of the immune response affect metabolism, but we don't really have a good sense of how the events of an immune response to a live pathogen actually take shape in a human, or in our case in a model animal. So what are the things that we think might be causing these metabolic demands? Well, first we have an innate immune response, and it manifests as fever and cytokine production, and these are fever raises metabolic rate, that's known, and the production of inflammatory cytokines, and you have an incredible movement from your bone marrow into sites of infection that is also a huge biosynthetic demand. You have lots of proteins being synthesized and cells being synthesized in this innate response. So we hypothesize that that's a large component of the metabolic demand. We also know that the adaptive immune response is incredibly biosynthetically demanding in that one clone of a T cell or a B cell can expand up to 10,000-fold to make an army to fight a specific pathogen. And so that should theoretically bring with it a significant metabolic demand. However, we don't really have a good idea of how those things fit together in space and time. And so for this, what we wanted was a system that is a well-characterized immune response to an infectious organism. And our favorite organism is Listeria monocytogenes. It's a foodborne pathogen. It is an intracellular pathogen, and it elicits a strong innate immune response, fever and all that, inflammation, as well as a very strong adaptive response. So with this response, we know the kinetics of it, we know the magnitude of it, and we can analyze how each of those components contributes to the metabolic effect. And so what we did was then to infect the mice with this pathogen and then monitor a bunch of different metabolic readouts using Prometheon metabolic cages. And these cages are fantastic in that they can collect body weight measurements constantly, oxygen and CO2 exchange. We can establish energy expenditure and activity and sleep, which I think is phenomenal. So you can basically keep track of how much an animal moves, how much it sleeps, and food and water consumption. All of these are really important to understanding our metabolic demands. And so this is what we know about the Listeria immune response. We infect at day 0, and here in red is the pathogen burden. So we know that the pathogen will multiply and sort of peak in numbers in the host at about 3 days, and then be cleared by day 7 generally, and that during this innate and during the pathogen period, you have the bulk of the innate response. So the animal will experience fever, lethargy, that kind of thing. And then slowly developing during this process is what we know of as the adaptive immune response. T cells are the major component in this infection, and we know that they expand logarithmically, and then they go through a period of contraction. The peak of that response is around day 8 to 10, 7 in some cases, and then you establish there's a contraction period in which many of those cells die, and then you store a population of memory cells that provide protection against subsequent infection. So what we wanted to do was ask the question of, well, okay, so we know all of these various things are going on at these time points. What's happening with metabolism and activity at all of these time points? So we used 2 doses of Listeria infection. The low dose is what you can think of as sort of a vaccinating dose. It creates an adaptive response without creating a strong illness response. And then a higher dose, this is really sort of right at a pivot point in which you get a strong adaptive response, but you do get signs of strong innate immunity, inflammation, fever, and signs of illness. So we wanted to compare and contrast those 2 things, being that the low dose would give us more of an adaptive response alone, and the higher dose would give us both innate and adaptive. So we first needed to confirm that we had indeed succeeded in these model systems, and this is the bacterial colonization. The high dose here gives us significantly higher colonization at day 3, which is nearly cleared by day 7, and the low dose is also cleared at day 7. And the animals experience very similar increases in spleen weight, and what that means is that is the extent of cellular proliferation that's happening in the adaptive response primarily, and that peaks, as I mentioned, around day 7 to 10, and it's not very different between the high and the low dose in this case. Well, what happens at the metabolic level is actually surprisingly different, but this is just confirmation that this is T cell numbers. This is just the antigen-specific T cells over time, and again, the peak is around day 7 to 10, and they're not dramatically different. But when we actually start looking at the metabolic impact of these 2 infection models, starting with body weight, we see a big difference. So our uninfected control is in blue, and these are younger mice, and they will gain weight every day as part of their natural growth. And so we put them in metabolic cages and we keep them there for 2 weeks, because that is the period of the immune response. And you can see that over this period, the controls gain weight incrementally. And then if we infect with the low dose of Listeria, we see a bit of a lag. They lose a little bit of weight, and then they sort of catch up. And that is around day 5. When they're back, they've recovered, and they're sort of not different than uninfected control animals. The high dose, however, those animals that had higher burden of bacterial colonization really have quite a dip in their weight, and they lose up to 10% of their body weight, which happens around day 4. And they begin to recover and regain weight, getting back to 0 basically at day 7, but they never quite recover to the level of control, and that we need to investigate whether that is maintained as a lifelong difference or whether they would recover later. But when we look at the metabolic associated effects of this, first I'll start by saying that the uninfected control and the low dose mice had almost no differences in any of these metabolic parameters. We looked at respiratory exchange ratio and at energy expenditure that's been adjusted for weight. And what we found was that at the early time points during infection, the mice that were infected with the high dose had a decrease in their respiratory quotient. And what that means is that they had preferential use of fat as a carbon source rather than sugar. The closer it is to 1, that would be closer to sugar as 100% of the carbon source, whereas the other mice, the controls and the low-dose infected, had very little change in that. The other sort of surprising phenotype was that the high-dose mice had actually a lower energy expenditure overall. And we know that metabolic rate goes up with infection and immunity, but in this case, the energy expenditure was lower within that first 5 days. And what's going on there? Why would the energy expenditure be lower? You'd think it would be higher. Well, it actually translates to differences in activity. So when you have a fever and you feel terrible, you generally stay in bed. And you're not doing that just because you think you're sick. Your body's actually telling you to not be active. And your body is actually telling you to sleep. So the activity percent drops way down in the high-dose mice, and the percent sleep goes way up. And so what we think is happening is that energy expenditure is going down primarily because activity is going down. And even though energy expenditure is lower and activity is lower, they are still losing weight. So the metabolic demand of this is actually quite significant. So I said I was looking for specific components of the innate or the adaptive response. Well, the timing of the weight loss and the changes in activity are really coincident with the innate response. They are not happening at day 7 and 10. They basically return to normal at that point, and that would be the peak of the adaptive response. And so it appears that the major metabolic demand is coming from the innate immune response, and that would be most likely pro-inflammatory cytokines and fever. So again, the most metabolically demanding response was happening very early, while the adaptive response was peaking later. And the other example is that mice that were infected with a low dose had a very strong adaptive response, yet they had very little change in their metabolic parameters. So and if you think about it, when you're vaccinated, you generally don't go to bed for a week, right? So your adaptive response is working very well while you continue your regular activities. So what we'd like to do now is drill down a bit more to determine which aspects of the innate immune response are really going to dictate this metabolic effect. Okay. And so that's what we're going to do is specifically point out what cells and cytokines are involved in this. And then eventually, we'd hope to develop therapeutics that specifically target these responses in the hopes that we could return people to work maybe one day earlier after an illness. So I'd like to thank the graduate student who's done most of this work in my lab, Robert Johnson, helped by another excellent graduate student, Adesola Olatunde. And none of this work would have been possible without Dr. Michael Green as a collaborator and the Auburn University Metabolic Phenotyping Lab, and Lauren Woody, his graduate student, who has been essential in setting this system up and helping us learn the ropes. And then I would like to thank the funding from the Good to Great mechanism from the Auburn University Internal Grants Program for this.